Welcome everyone to the latest DRIIMB webinar. Just some background for those that are new to this. DRIIMB is a consortium of several collaborative computational projects in the area of structural biology, specifically covering crystallography, cryo-EM, NMR, and biomolecular simulation. And the aim of this is to look at integrative structural biology. Now, part of the wider aims of DRIIMB are to reach out to neighbouring communities and look at complementary information that can be brought to bear on biological problems. So it's in that spirit that we're very happy to welcome Frank Sobott to give the webinar today. Frank is chair in biomolecular mass spectrometry at Leeds University, following a previous career at various places, Frankfurt, Oxford and Cambridge with Carol Robinson, who I guess many of us know, Structural Genomics Consortium, Antwerp, and now at Leeds. So I think we're all vaguely familiar with mass spectrometry, but maybe get lost in the details of the various different modalities. So I'm looking forward to the talk where Frank is going to explain everything. Thank you.
Thank you very much, Martin, for the kind introduction. And I've not managed to explain everything, but certainly some interesting aspects where we're at and what I think could be future directions for some of these techniques which we group together under the term structural mass spectrometry and how far we can achieve some integrative modeling with them. So I had, even though you introduced me, which is very kind, I had very briefly put up some points just to say why I have an interest in this, because I'm not really someone who does much modeling myself, but I was involved in early developments in native mass spec and also with Carl Robinson. And I guess I learned a lot about how different techniques can work together in a structural genomics consortium. But I also chaired a so-called cost action as an EU-funded kind of training network, really. It's not funding research, but training, best practice, also white papers in different fields. And this was based on, I guess, the structural MS techniques. And it ran for 4 years. And actually, we laid some groundwork, particularly strongly with the UK community, but I would say European colleagues as well, and some international ones on trying to get more out of our data. And at the moment, I'm still involved in Fragment Screen, which is a big project also led by Harald Schwalbe, who leads Instruct Eric. And I lead the mass spec there. And the idea is that we also want to see, even though it's sort of specifically targeted on ligand binding, but I guess more generally how we can use mass spec data in a more thorough way and make it more accessible as well, deposit it properly. So what I like to say, slightly tongue-in-cheek, of course, and like to sort of challenge people with, is also that we want ideally, and not in all cases, but in many cases, we want to understand ensembles instead of just a single structure, so sets of structures maybe, but also we want ideally to be able to make a movie rather than just get a static snapshot. And one of the sort of enabling ideas of good data repositories and also best practice in recording data and all that, what I guess happened for many of the other techniques years ago, is that machine learning will be enabled by very good training sets, but also by a good understanding of what that data really means, because it does depend on the experiment, probably a bit more than in some other scenarios, such as X-ray, where when you get a crystal, you kind of have a structure, but in NMR, you have many flavors of experiments, and mass spec's a bit more like that. I also still think, and this is maybe also meant a bit tongue-in-cheek, that there could be a role for something like a CCPMS at some stage. So I want to, I don't think I have to convince this community, but I want to quote Freeman Dyson, who did emphasize, and I think we have to repeat that sometimes also to our funders, that new directions in science are launched by new tools as much, or maybe even much more often, than by new concepts, because a concept-driven revolution, I mean, it's probably a bit biased towards tools, I should say, but is to explain old things in new ways, which can be very important. But the effect of a tool-driven revolution, so if we really enable new structural methods, for example, is to discover new things which we kind of didn't even know about yet, I guess, and which still wait to be explained. So I just want to really fly the flag a bit also for methods development And I'm equally interested in applying methods for sure, but I think this is sometimes a bit neglected. Now, this is a very trivial slide. It's even from Wikipedia. Of course, we know about primary to quaternary structure of proteins, but I want to use this a bit to map mass spec onto it. And of course, we are well aware, I'm sure all of us, how proteomics, denaturing mass spec is used. Bottom-up proteomics, when you have gel bands, you identify an unknown protein, you identify PTM in a biomarker in the blood of a patient. But intact mass, where you don't digest, but you still denature, is very important for quality control, for example, for antibody, biopharmaceutical products, and also oligonucleotides, many other things. So there is, of course, a lot of mass spec done, which is maybe a bit further away from structure. Although I should be careful that even bottom-up proteomics in various ways, I'm not having the time or don't want to overburden you either with detail, but there are ways where you can also get quite interesting structural data from readouts, which are entirely tryptic peptides ultimately. But higher order structure is what it's more about. So what we classically call 3D structure. So structural MS, there's basically two types of methods, and I will only focus on the 5 most important here today. The native methods, native MS and immobility, where we still work with a native buffer and we don't digest or denature, but we kind of read out directly from a solution what is in it. But there are labeling methods where we have the kind of labeling, which I'll come to, of course, in a moment, labeling or cross-linking, where we modify the protein in the native state, but then we can actually, because the label hopefully stays on, we can then process it a bit more like a proteomic sample and find the labels and reconstruct what the structure was like. But there are interesting points which are also a bit more peripheral to the topic today about how well we can deal with several states being present, conformational states or binding states, maybe 50% is only modified with a phosphorylation. How can we not just deal with that, but can we even quantify this a bit rather than being selective and maybe characterize one of these states and be a bit blind to what else is there? Now, that has to do with how far different techniques are capable of dealing with, I guess, purified protein, which is classically what also structural biology does, but I'm fully aware of tomography, for example, versus can we go potentially into cells? I mean, that's always the best if you can, right? And in addition, in vitro, in vivo, in addition, also can we potentially even spatially resolve if you go into cells? Can we see the same protein in different states? We like to work a lot with colleagues on amyloid aggregation. And if you look at a brain slice from a patient, then there's a lot going on, but we might be very selective with cryo-EM when we see amazing fibril structures, but we don't really know that much about what else is there and in what is the microenvironment of that scenario. So just to motivate the dynamics and the sort of snapshots in the movie, I would like a bit more. I mean, this is kind of inspired, borrowed from Arvin Pearson, who's in Hamburg. He used to be in Leeds, where she likes to show conformations of a horse, where apparently rich gentlemen were arguing forever if the horse ever has all legs up in the air, and then high-speed photography eventually came to the rescue. But obviously, we have many proteins where structure informs function, yes, but dynamic structure. So it can be motion in the transporter, at least it's 2 states, right? So we often challenge ourselves in getting more than one structure, and cryo-EM is really getting quite good at that these days. NMR inherently can, of course, also look more at dynamic aspects. So that's an aspect for mass spec I will bear in mind, and maybe we'll come back to it later. I did mention the big 5, the 5 methods. So that's the 2 intact or native analysis methods, native MS and immobility, and come back to those. And there are the 3, I already mentioned them, 3 major approaches for what we could consider labeling in the widest sense, where we do some chemical modification and read it out at the peptide level. And that's basically the structure of the next, hopefully about half an hour maybe, where I will give you some examples, but also specifically try to address the current status and challenges we have in how we could get much more out of this data and how we could start to model more routinely, either select a model from a pool or even drive modeling. But I want to briefly say why mass spec. Of course, integrative structural biology is very much about not doing one or the other thing, but maybe the right combination of things. And mass spec methods can border on proper structural biology. They can easily be classified as biophysical methods. So we don't have to argue the semantics, but it's a whole range in which they can sit. I think one strength of structural mass spec is that rather than just detecting maybe a sequence variant, a truncation, a mutation, a PTM, you can in principle detect the complex, but see that this complex has a phosphorylation, and only when it's there, the other protein binds. So you can do that in a heterogeneous background. You can link sequence variation heterogeneity with a conformation potentially, but also with the interactions with the complexes it forms. So I think that's an asset here. So dealing with heterogeneity in general, if you have different conformational states, we work a lot on alpha-synuclein. It's an IDP, of course, so there isn't really any defined structure. And only when you offer it some lipid bilayer negative charge on it, then it might form a partial alpha helix. But you can kind of see the unstructured states as well as the structured states kind of side by side. And that's quite unique in a way, at least for other structural techniques. Of course, some biophysical techniques can do that too. And then you can look at partial assembly intermediates, oligomerization as well, even if it's quite heterogeneous. In addition, intrinsically disordered proteins, really like a third of the proteome, depending on where you look, roughly lack some structure at least some of the time, which means we would like to probably know more about them. I guess you could think no structure, no function. I would be careful there because a disordered protein can also adopt a structure, and we just need to understand how it switches basically into these states. It is fairly unbiased. We don't need any protein-specific labeling. Even if we say we have labeling techniques, we don't need to introduce a fluorescent label or something like that to a specific position. So we can discover things we weren't even looking for. And we can actually get thermodynamic stability data and exchange kinetics. You can, for example, even look at protein dimerization, homodimer. But if you have half your protein labeled like you would for NMR and the other half not, you will see how the label scrambles in. So you can actually get the kinetics of the dimer interface that way, for example, in thermodynamic stability. You can get pseudo-KDs. I can later very briefly mention, of course, there's an experiment which happens in a gas phase, so it can distort a little bit, but we can control that and what happened in solution. But you can also compare relative strengths of interfaces, for example. You can do competition experiments, that kind of thing. And the frontier for us, and there is some success in some areas, is that we really want to work more with complex samples. In theory, we want to do everything in vivo. Of course, in practice, that won't always work. And we want to develop approaches where we can maybe even do spatially resolved mass spec that can be that we extract in a spatially resolved way, or that we even try to label it directly in the tissue and then extract after. So I think that sets hopefully the scene a little bit and motivates also why mass spec even appears in a sort of more structural, I guess, background. And it's not just a biochemical detection tool clearly at that stage, even though a lot of classic proteomics is doing that and it's very important. So I will start off, like I said, a big five with a native MS approaches. And what we use is basically a native condition in which the protein is in a native buffer, like maybe on a native gel as well. So you want to preserve your non-covalent interactions. That means conformations, but also interactions complexes. You can get data on binding strengths, ligand screening is possible. You can look at assembly-disassembly pathways and intermediates, and you can also look at disordered unfolded states. Here's a sort of simple representation of the same protein, more folded and more unfolded. This is an electrospray ionization, so you have mass over charge. You have a bit of dimer, but otherwise all these other peaks correspond to the same monomeric mass. But how many charges you get in electrospray, it's protonated, depends on the surface area. So you actually get an idea already, it's bimodal, and that you have multiple conformations, just even from the charging. But this is not as highly resolved as ion mobility, which I'll come to in a moment. But you can look at structural ensemble. So you see coexisting states, monomer dimer, and also folded, unfolded. It's really relatively unbiased. I mean, your detection is not so much depending on if it's structured or not. It might just be depending on sensitivity in the mass range and something like that. So heterogeneity is fine. You actually see coexisting states, not just the one you can characterize. And yeah, you can deduce something on the solvent accessible surface area based on charging. So there's actually an interesting field of research where people use that to predict kind of folding states. Traditionally, we need to use volatile buffers, which means we replace whatever pH condition, of course, we can match, but ionic strength, we would like to use normally ammonium acetate, which then replaces, let's say, sodium chloride from a gel filtration. But more recently, we and others have shown that sub-micrometer class needles. So when I show a picture in a moment, when you think about how nanoelectrospray mass spec is done, we use often static spray. We actually use patch clamp-like glass capillaries we can pull. And with a laser puller and with the right set of conditions, you can get really accurate so-called nanopipettes. They're even used for, I guess, a single DNA sequencing and this kind of stuff in other environments, but they also work as electrospray emitters. And then you can tolerate a lot more salt. So we more recently find we can also work in PBS buffers and these kind of things we don't normally like so much. But traditionally, we work more or less with isolated purified protein or complex. It can, like I said, assembly intermediates. There can be some heterogeneity, but you wouldn't really want to work with a whole cell extract. A colleague has shown, though, Michal Sharon in Israel, that if you overexpress a protein, it's the most abundant protein, you can actually even work with a crude lysate and still do nano-electrospray, native electrospray from such systems. So I think there's a boundary we can push a bit. And the instruments, they used to be much more dedicated that we asked manufacturers to modify them, mainly because of the mass range and much larger particles than typical peptide work. But a lot more instruments now already accommodate that. So you will find a commercial instrument for most manufacturers which can deal with a lot of this now without having to get working on it yourself and change it a lot. So very briefly, electrospray ionization is key to native MS, like it is also to peptide level analysis. So John Fenn got a Nobel Prize for it. You use this sort of capillary, or you can also do a pump. You can do a constant flow setup, but for native MS, it's more typically static flow. And of course, there are also more automated systems. The idea is that your ions, which are of course still in solution at first, they kind of dry up, but they're kinetically trapped when they come out of the droplets, so that the structure does not really adapt ideally to the new vacuum environment. So it happens quickly enough that it preserves its folding state by and large, at least to a large extent, because there can be very small differences, like a lysine can self-solvate, you know, the lysine group can move a bit and these kind of things. And it is also important to say that if a protein isn't stabilized, so it's not folded, it doesn't have salt bridges, it's maybe a full IDP, then the question becomes a bit more how gentle you have to be to preserve that. But actually, we have very good evidence from alpha-synuclein when we did a systematic study comparing to NMR and SAX data, that even the extended disordered states are preserved in this process. So the ions are released in such a gentle and fast way that they're kinetically trapped more or less in their solution folding state. I think that's the premise of native MS. And then very briefly, maybe just a summary slide. If you denature your protein complex, maybe with two ligands and it's a dimer, you get these highly charged, this is without digestion now still, these highly charged peaks, but they do give you information on the sequence mass, the total complement of PTMs, not necessarily where they are, but how many you have and where they are. The proteoform basically, but also the subunit composition if you have a complex. So taking it apart, looking at the pieces is interesting, but then in native MS, you keep it together. You're looking at the whole non-covalent complex preserved. You can see ligand binding, also heterogeneous binding. You can do ligand competition experiments, subunit stoichiometry. And with ion mobility, you can look at conformations, folding, unfolding. I'll come to that in a minute. So one example I like to show this, it goes back quite a long time, but it was even in collaboration with Aztecs, the fragment discovery company in Cambridge, in the late Chris Abel. We just looked for the first time, I guess, at how much we could get close to an actual solution KT and mass spec. Now, I would always be careful because you do your binding experiment by titrating NADP to your enzyme in a solution, and you see peaks shift from the apo form to the ligand-bound form. And if you do that for many more steps, you can make a proper binding curve and get a pseudo KT for mass spec, which actually was same order of magnitude as an ITC. But I need to say that a very hydrophobically binding ligand really doesn't have much of a barrier to fall off in a vacuum, right? Because you don't have any hydrophobic effect really in the vacuum anymore because you're not competing with any solvent. But the point again is it's more or less kinetically trapped. So if you're careful in many cases, and if you want to maybe rank ligands on a relative scale, this can actually go quite far. If you want a KD, you shouldn't use mass spec, but you can get quantitative, semi-quantitative binding affinities. And that can be interesting for things like also the fragment screen or the sort of efforts people are doing to high throughput some of these methods more for ligand binding studies. Obviously, that's happened for crystallography, certainly at DRIIMB, but for QIEM, it's still more or less one ligand, one grid at the moment. It's also not cheap. So how could we allow for more high throughput readouts, not just binding yes or no, but maybe some additional information, stoichiometry, targeted protein degradation, where you then change the stoichiometry when you bind the ligand. So you actually go a bit further than just saying it binds. And so mass spec is going to have more future in that field. And I should say that companies like AstraZeneca, together with the Medicines Discovery Catapult, have also worked on a very fast readout, acoustic mist ionization up to 3 ligands per second. And we've had a case studentship with them where we showed that this works for non-covalent binding. So actually reconstructing this whole setup and making it more robust and more user-friendly, I think there are clear opportunities for high throughput mass spec-based ligand screening, which then might have a readout even with ion mobility where you see conformational effects. And as I said, it could also change the stoichiometry of a complex. So if you just want binding yes, no, maybe not mass spec, but there are many other scenarios where this, I think, has some real potential. I want to show a couple more examples. This is one from Cal Robinson's lab with Sarah Teigman, where they actually used something not so much inside the mass spec. They used just classic disturbing solution disrupting conditions, perturbing conditions like a co-solvent. And you kind of weaken a complex and it falls apart into subcomplexes. And then you can see the sort of basic unit is maybe the dimers, so the hexamer is a trimer of dimers. But in this case, Sarah's angle was that the evolutionary intermediate, she did obviously look at some other genetic information, kind of matches the destabilizing reverse pathway in an experiment you can do in a native MS condition, where you just do it in different solvents and you see different assembly states. So there's quite a lot you can do by just using a smart solvent setup and then reading it out with native MS. So I think it's a nice example for not just characterizing an oligomer, but understanding a bit about the evolutionary history. And you can, of course, have a subunit which is maybe got some mutations and see if it still forms the same amount of hexamer, or maybe it's just partial. You know, you can, that way you can also get thermodynamic information. So Justin Benesch has done some nice studies with small heat shock proteins where he actually explored that a lot further. And yeah, there are papers then I would appeal to people to look at. I don't really have the time to go into too much detail. I just want to highlight some aspects here. But you can measure relative stabilities. You can look at subunit exchange. If it's the same subunit with the same mass, then you need to label it with mass, which means isotope labeling. And then even though it's chemically the same, you can still see the mass shift, how it propagates into the oligomer, for example. Another example, and this is something company Thermo, of course, Thermo Fisher Scientific, we all know from DRIIMB, but also from mass spec in particular, has pushed a bit, but we were actually beta testing that together with them. It's a more automated native MS with an online buffer exchange. So you try to use some kind of miniaturized gel filtration approach because you need to get rid of these volatile, non-volatile salts and purify your sample a bit. And the idea is that you put that through the mass spec, you get an idea of the heterogeneity, also depending on what complex this is, how it might change over time. And then the moment you actually put it on a grid for DRIIMB, you kind of align the mass spec compositional data with a hopefully high resolution DRIIMB data, but it might still not show some more dynamic parts of the sequence, or because you have that heterogeneity. I know with Relion it's become a lot easier in DRIIMB to maybe see different particles on the same grid. But I think the idea is it can still help quite a lot to know what to expect and what's going on. So this is something where Thermo as a manufacturer has sort of built some software where this data can at least be captured together. And the idea is to then also utilize it and potentially also for modeling. But first of all, to just really have a more thorough characterization of a sample. When I worked at a structural genomics consortium, we had a rule that nothing goes into crystallization, which hasn't got a proper mass spec with it, because very often you do find unexpected truncations or mutations. And then I guess your X-ray reconstruction suffers from using potentially the wrong sequence, even if it's a minor thing, maybe. Then another point, and again, just to give you some idea what the diversity of approaches is. So if you do native MS, you don't necessarily have to keep it all together. Maybe you do that first, and then you might want to take it apart. But rather than in solution, you can do that in a mass spec. And one approach has been developed mainly by Vicky Wysocki, a colleague in the US, is called surface-induced dissociation. So the typical sequencing of a peptide is done by collision-induced dissociation. Here's the ID on the left, where your peptide goes to the gas, it has many collisions, it slowly heats. You can really consider it like a heating, just in vacuum. And it will break in the weakest bond, which under those conditions is a peptide bond. So you get your sequence segments. But if you do that with a non-covalent complex, you actually, hope you can see this a bit, you're kicking out one subunit which unfolds. You're so asymmetrically disrupting the complex. The reason is that this is charge driven, so it kind of melts, and then one subunit gets ejected with a lot of the charge. If you do, however, SID, so you bounce it really just once, more or less, against the solid surface, this is much, much quicker. You still impart a lot of kinetic energy, converts to internal energy, but it doesn't have time to melt. It kind of just explodes, you could say, right, hand-wavingly. So you get a near symmetric splitting of your complexes, and this becomes quite informative for larger complexes, for oligomers, for example. And you can get an idea really more of the architecture, while here you just kind of eject one subunit and you don't know so much about the rest. Another approach is electron-driven. I know it's a bit more technical now, but you can also, in some commercial instruments, and we've collaborated with manufacturers before, you can do electron transfer or different flavors of electron-driven dissociation, where you basically, a chemist would call it homo-LUMO interaction. You activate a peptide bond electronically. You get a different type of fragmentation, but it's very gentle, and it's only in a backbone. So your side chain contacts, your non-covalent interactions actually survive, even though you're cutting the backbone. And then you can get information on the solvent accessible surface area, because that's where this reaction occurs, even in a native structure. So it's quite interesting that even the fragmentation you do, starting from a native experiment, can be informative. And recently, so this is from that paper, again, I would refer you to it, and happy to share the slides as well later. Stefan Lindert, who works with Vicky Wysocki a lot, and worked with Jens Meiler, who I mentioned later before. It's interesting that he is really one of the few people who's pushed attempts to model or to improve modeling with native and ion mobility data. So he's been working with Rosetta. I guess like Jens Meiler worked with David Baker before as well and did the ligand binding there, and come to that later. So when you have SID data, you can just refine your sort of pool of possible structures, and you get better RMSDs by having this knowledge of where the interfaces are from how it's being disrupted in the SID experiment. So I think there is some, but this is really quite a singular story so far. There is really some insight into how this can work. And I would refer you to the paper for more detail. I'm happy to take important questions now. Otherwise, we can obviously reserve some time at the end. But if you had a question about native MS so far, we could do it now. If it's easier, I'll continue. And you can take a note for later. Because I was going to talk now about ion mobility, which is very much based on a native experiment. So we're still in the same sphere of mass spec. But what we do here now is a bit like a native gel, but obviously in a gas phase. So we have an element inside the mass spec where your native conformation or oligomer, whatever it is, complex, goes to a cell which has gas in it, but not to activate the ions so they disrupt and fragment or unfold. But it's very gentle. It's actually just to have viscosity. You could call it viscosity. And that way you can then measure the time they take depending on their cross section. So I saw Eric
I
had
think
a
we
hand
have
up.
a question. If
Yeah,
do you
Eric?
want to take a question?
Yeah, I'm happy to take the question. Hi,
So,
Eric.
Eric?
Hello, Eric. Can you please post the question in Q&A panel?
I'm not sure. Do people have permissions to unmute or? If not, if you put it in the Q&A, I can read it out.
We can discuss it later as well if it's not easy to activate a microphone now, but I've taken note of it. Thank you. So in the ion mobility experiment, we are extending the native experiment because we now have a stage in which the ions are measured according to their size, to their collision cross section. The way we should regard that is that the ions tumble very fast. So if I have an object, I just, I don't know, take my badge in case you can see it, it's of course a very flat object, but it's rotating fast. So we are looking at the rotationally averaged shadow of this object, not one specific orientation. So these ions are not aligning. This is a weak electric field. It's not heating the ions. It's just like, I like to say, like when you parachute jump, I don't personally, but when I trained once in a doctoral school, and someone said she had done 3 more out of 280 she done in the last 5 years. So obviously when you open your parachute, your mass doesn't change, but your collision cross section changes a lot. So your drift speed towards the surface of planet Earth is a lot slower. And actually here as well, if you open up the structure, it will be slower, but you reach a drift speed. I mean, there's an electric field which accelerates, but with a friction, it's an equilibrium. So you have relatively quickly a more or less constant drift speed. And that relates to the collision cross-section, to the overall extension of the particle. That's interesting for folding. It's also interesting for different alternatives arrangements of subunits, like a topology of a complex. And it's very interesting for intrinsically disordered proteins because you get more conformational insight than purely from how they charge with a solvent accessible area. So it's a great way to look at complex ensembles. It's really quite unbiased again. It doesn't matter if it's folded or unfolded. It has a collision cross-section. Some of you might be thinking about SACs now. It's really quite close to it. Of course, it's in a gas phase, but we do have, well, maybe I come to this. We have a collision cross-section here. We do have a single size parameter. What I mean by that is we replace it with an equivalent sphere, you could say, while I guess SACs has more of an equivalent ellipse-shaped, egg-shaped object, right? So it's one size parameter, but clearly quite powerful if you compare folded, unfolded. It is still a native experiment though. So what I said about salt tolerance, which is now got a lot better, but still a limitation. The resolution is relatively low, but we think sometimes 1.5% difference in collision cross-section is relevant. And it pretty much needs a model to interpret. So we can make candidate models and we can say, if it looked like this, would that match or not? You know, it's much harder to derive a structure just from one single parameter. But one of the things we are keen on is maybe really teaming up more with colleagues in SACs and learn from them a bit more what we could do in I-mobility. So the I-mobility cell, just to repeat, ions go through here. It's an example of a topology of a pentamer. But it's important to emphasize now, we still have mass over charge. And now we have an additional dimension. Here it's an arrival time, but if you know some proteins, you take some proteins where you know the collision cross-section, you calculate it from their PDB, and you take well-folded proteins, then you can translate that into collision cross-section in square angstrom. And it does still depend on charge. That's why it's not constant. So the arrival time still depends on the charge state. And these are, again, different charge states of the same pentamer. But you can actually see if there's any heterogeneity, if, for example, the peaks in between, if they sit on that line, or if you have a second population. And this is really useful if you have a complex, maybe self-assembling amyloid-like samples, for example. So it's like a gas phase electrophoresis. You move the ions through normally nitrogen gas, but you can do all sorts of things. You get a SACs-like shape parameter collision cross-section, but importantly, you get collision cross-section for every single peak. So if you had a pentamer and some assembly intermediates, and we've done studies where we had like maybe a tetramer which was linear and not a circle, then it would not lie on the same trend line either because it's more extended, right? So you can get trends quite quickly, even if you don't calculate a model. But it's based on something really old, 101-year-old now, which was the idea that if you have an object and you turn it in all directions, you get these kind of cross-sectional areas. This was published in 1925. Actually, next month, we have a celebration in the US for 50 years of ion mobility. So the technique goes back quite some time. It's also used in the scanners, not in the X-ray scanner, but if you get secondary screened at an airport with a wand, then it's an ion mobility machine, but there they just look for nitrogen-rich compounds, small molecules, explosives, drugs, and so on. So it's a technique which is not that novel, but a combination with native MS is really quite powerful and relatively recent. Right, so one example, briefly, I want to also show an oligomer structure because I think structural biology has got lots of new challenges now in oligonucleotide space, not just in a protein space. And one well-known example is the cocaine binding aptamer, which actually binds quinine better than cocaine. But when you look at the canonical one and you look at the stem loop and you try to understand how the binding site is formed and why they're so highly selective, they're actually as high affinity and specificity as antibodies can be. So they're also very interesting drug formats. But then you can ask questions like, is it a sort of topology? Is it a specific H bonds? What difference does a small change, for example, here make? And how important is it if that stem in the sort of simple structure you have here, if that's a bit extended, which makes it, of course, more stable. So when it's short, it's more open because it's not stable double strand yet. And these two variants have a more stable preformed double strand. So can we use mass spec to learn something here? Of course, there's NMR data, which was very useful. The first thing is if you randomize the same sequence I showed, you really get no binding of the quinine ligand. So it's not just some electrostatic non-specific interaction in the gas phase. But then if you take the canonical sequence and take these variants, so if you have a small change away from the binding site, it does little for the binding affinity. But if you take the pre-shaped sort of stem, that actually helps to also pre-shape the binding site. It does improve the binding, so you get more of a boost in KD. So far, so good. But when ion mobility comes in now, this is now only done on a relative scale. So we're not looking at CCS, we're looking at the moment I add a small molecule, how much does the global conformation change of the aptamer? And you can see that there's a relatively big change for these two, which were the weaker binders. And the stronger binders really had kind of the opposite trend. Now, what that tells us is that there's an entropic penalty for having to rearrange to actually properly form this double strand here, which goes against the affinity of binding, and it's shown as a bigger change in ion mobility between apo and bound state. So you can infer some qualitative thermodynamic data just from such a comparison. That's what this example is meant to show. You can do something like that with proteins as well. Another brief example, just to show you some ideas where we can deal with intrinsically disordered proteins. It's a recent paper where we looked at alpha-synuclein and specifically at metal ion, but you can also look at other interactors, and EGCG epigallocatechin gallate is a famous sort of modulator of fibrillation as well. And of course, we're interested in understanding what happens even at the monomer level, which leads to a bit maybe a different polymorph or different downstream fate of how this protein behaves. So in native MS, you can get very complex spectra, which really correspond to many coexisting conformations. If you then go into ion mobility, you get these profiles, which you can deconstruct into components. They're showing even more detail. This is only shown for one of these charge states, and there are such profiles for each charge state. So the ensemble represents everything. But this is just graphically showing how you shift without necessarily being able to put a detailed structure next to each one of these collision cross sections, but you globally shift your ensemble. And this really correlates very well with differences in the kinetics of aggregation and also with the downstream fate of amyloids. So it's an interesting question what mechanistically happens, and we don't fully answer this yet, but we've used recent modeling tools like Calvados from Kristen Lindorf Larsen, based in Copenhagen, to try and at least get an idea of what these different sort of subsets of conformations can correspond to. And we also did NMR here, chemical shift perturbations. So again, it's a paper maybe to look at. So just want to, I guess, start to wrap this up a little bit. So I think it's a bit about ensemble structures here, and ion mobility data can be matched to calculated CCS of models. Of course, the question is, especially for an IDP, where you get the models from. But you can always ask, assuming it looks like this, what would I have to look for in my ion mobility data? So you're free to do what you want there. I should say that folded proteins, which we also use for calibration, they match the CCS calculated from the PDB model really quite well. And that's also the basis of knowing that we are not destroying these structures when we release them in electrospray. But we deal with like broad spectra showing the whole diversity of IDPs from small to large, depending on the protein. And that's, of course, a challenge for modeling. There are new tools, also Rosetta, and I think actually, sorry, not so much Rosetta, but Martini comes out with like more ensemble type modeling. And I think what's important to say is even if we don't do that, like I showed for the aptamer, if we see a shift, if we see a change, we can actually consider that shift on a relative scale as a meaningful way to see what effect does a binder have, for example. This is a very recent paper I just picked up again from Stefan Linder with Vicky Wysocki from the US, which I was quite impressed by because they're now even specifically using CCS data and again, seeing how AlphaFold and Rosetta can be helped along in deciding what actually really happens rather than what's possible. So summarizing for native methods, which includes ion mobility, direct detection of native structure and interactions, non-covalent interactions have to be preserved in mass spec. That requires careful tuning and also a certain type of instrument. Then you get composition, ligands, stoichiometry information, size, shape, topology information. You can infer folding conformational ensembles, but it does really ideally need to be underpinned by at least proposed models. So maybe you can score existing models rather than drive modeling. But it deals quite well with heterogeneity. It can also deal with like a lipid effect on a transporter. Doesn't have to be an actual ligand. So there is a lot of more complex structural biology where mass spec can help a little bit, I think. And since I also in my title talk about how we can be better with data as a community, the cost action we had led to quite a few standards papers, and this was more driven by the mostly UK community in this cost action I shared. And we had 2 papers back to back where we looked at, maybe it needs an update after more than 5 years, on software requirements for native and ion mobility, but also what current computational strategies and challenges there are in terms of modeling, in terms of, I guess, using data to confirm a model you already have, right? That's what I call a bit of a selective view versus really using it to see what is there and is there something I didn't expect, which we don't always do. But problems are still there. Most papers which are published, and that's not necessarily terrible, but they should normally have some kind of a technical replicate. So you measure your mobility in more than one way. And you can argue if you do your DRIIMB structure in more than one way, but of course there are inherent quality parameters also in any structural method. So I would like to, I guess, get the community to be a bit better, and also statistical analysis, how big a difference is relevant, you know, that kind of thing. Again, selective perception can mean they see what they want, and they happily report it and don't care any further. So best practice also in how to tune your instrument so it's actually truly native, and how far does it depend on the instrument is important. Data metadata reporting does not happen normally, only in the paper itself. Machine learning enabled depositories don't really exist. We are talking a bit to EBI Pride, but we are currently actually in an effort with a US colleague and also Pfizer US, in proposing at the next big conference how a metadata capture, which we even want to automate from the instrument, how it could look. We are talking to Instruct, Eric, and Fragment Screen, how we can tie that into existing software infrastructures, because if you don't even have more than a zip file where people put their raw data and some text in a paper, the machine learning is going to be very hard, right? So we want to enable that machine learning, secondary use of data, fair data. As a community, obviously. I come to the second part, hopefully take a bit less time and stay within the hour. HDX mass spec might be quite familiar to some of you. It's a native labeling technique. The nice thing is now we are not keeping it native beyond when it's labeled, then we quench, then we digest. But you can label even in, let's say, lipid nanoparticles in complex samples, like a SMULP particle. You can try it in vivo. That's not straightforward, but there's a real push for that now. If you have a high affinity binder where you can fish it out quickly, then you can try to do the labeling in an extract, maybe from a cell. It's become a really key technique in all the epitope binding studies, not just for antibodies, but also for nanobodies, for adherents, affimers we have here in Leeds, even for aptamers. At least you can get the patch which is protected when something binds quite quickly. And this has become very important for pharma industry. You get allosteric effects in kinase studies, for example, that's quite important. You see effects where you don't expect them. IDP dynamics as well. IDPs, intrinsically disordered proteins, hard to study. So structural transitions, because we are in a strict way, we are not really looking just at one structure, we're looking at a fluctuation of a structure. That's an asset though, we just need to learn how to use that data. And we can do equilibrium experiments, which is normal. So you have your ligand, for example, or your binding partner, your antibody, antigen, already preformed, and you're not looking at the exchange of these subunits. You're just looking at the exchange of the label. But you can do a non-equilibrium experiment where you add the deuterium together with the ligand. Then you get at least 3 things. You get kinetics of binding, kinetics of the structural effect of binding, and the actual kinetics of HD exchange. So it becomes a challenge how to deconvolve that complexity. But J.J. Phillips in Exeter has a nice paper out where he's shown how to do this, and also Derek Wilson in Canada. It will become more routine. There will be a commercial setup for that soon on the market. Typically, you still work with purified protein. I guess that's fine if you make antibodies for a living as a company. Then it's also a QC step. Is it aggregating or not? But it is, in some structural biology scenarios, is still a shortcoming. It is actually, let's be honest, it's quite slow and expensive. It takes 1 day per condition. So if you want to compare 3 mutants, it takes you a better part of a week, and it's taking that to do the experiment and twice as long to actually analyze the data. And that is quite irritating because it just needs a much better software workflow, which could be done. Bits and pieces are actually out there. At the moment, HDX is very powerful, but it's only relative. You always compare 2 or more conditions, and it's only qualitative. You say it's different with the proper statistics at least being done, I think that is good practice now. But you could make it actually absolute. So you really measure exchange without comparing two scenarios. And you could make it then quantitative. And there are some shortcomings why this is not happening, which I can also say more about. But the other problem is if it's heterogeneous, if you add a ligand and it only binds 50%, then your answer is going to be a mix of the two states. So you better have something you drive to close to completion before you do an HDX experiment. For those who don't know much about it, I can very briefly show how it works. So if you have a protein and a protein plus some other compound, you will have certain areas, if you can see it, where there are no red dots, where the deuterium can't get anymore, because exchange happens on the solvent accessible parts, but more importantly, where there is no secondary structure. So where the protein likes to H bond to water, to solvent at least, rather than to itself. Those are the exchange sites you will see. That's important to emphasize. So it's not predominantly solvent accessible surface area. It's actually more secondary structure we are seeing here. But that's great because there are not so many techniques which can do that. And when you've done your labeling and you waited for a predefined amount of time, typically 5 time points, you can see it here as well, or maybe 3 or 4, then you can quench. So your top pH, your top temperature slows it down massively. Doesn't stop it, but it goes down a lot. And then you quickly digest with a protease, an acid protease like pepsin. You quickly separate your peptide, so you can't just store it, you have to do it inline. And then you see this shift in the isotope pattern, which is deuterium incorporation. And the shift itself is actually meaningful, but it's very often not looked at, but you look at the difference plus minus a compound. And you see how the shift slows down, but you can locate it to the peptide in which it happens. So you get peptide level protection data, and you get these kind of curves where protection means less exchange at the same time point in the presence of a compound. You can highlight where it happens. But you might find an unexpected acceleration elsewhere because maybe that secondary structure is now gone. So that's where it can get really interesting, but also a bit of a headache to know what to do with it. Don't want to go through the fundamentals. I want to again show a really nice article I can recommend, which I think is one of the better ones, or best ones, in fact, to read. But I want to emphasize that the kinetic constant of HDX, of exchange, is a sum of 3 components, but the base catalysis is the dominant one at normal pH. So your OD minus concentration, not OH minus, OD minus concentration in the solution determines how fast you're exchanging. And of course, the structure locally on the protein determines how fast you're exchanging. But there are several factors coming together. And the so-called Linderström-Lang model, Linderström-Lang was working for the Carlsberg Brewery, Carlsberg Labs later in Copenhagen. That assumes there's a closed and an open state. So closed means exchange incompetent. Open means exchange competent. Of course, this can fluctuate locally, depends on secondary structure. And then the exchange would be from the open H to the open D state. But often people neglect that part of the equation. So actually, this is equally important. And in fact, this K close and this K close is often assumed to be the same. But we all know about isotope effects. So we are in a position now to start and discuss how to modify this model a bit to be more accurate. But typically people do 3 to 5 time points, but then they take the one where they see the biggest effect and they throw away the rest of the data. Not great. Actually, Ollie King in Oxford's recently started to publish some interesting work together with GlaxoSmithKline where they use the time evolution of these curves and get more information out of it. But you need to get your stats right, obviously. I said already, it's usually in comparison, differential and qualitative. For epitope mapping, that's all you need. It's perfect. But it could be absolute and quantitative. It could drive modeling. So then you could model ensembles potentially even because you can calculate to some extent what the HDX would look like for any assumed conformational model. But it's much harder at the moment, there are attempts of course, but to deduce that kind of information just from an HDX pattern. Some standardization is underway. I mean, there's one paper I'll show in a moment, and we're actually working as a community on another one now. Challenges remain. So there are some attempts, successful attempts at ensemble reweighting, where people look at 2 possible states for GPCR, for example, take HDX data and then say, okay, based on HDX data, I can say how the 2 states are populated. So in a more limited set of possibilities, there is already some interesting modeling you can do. Can be used for non-equilibrium experiments. I mentioned this, but not typically. You can get KD information. You just titrate more and more ligand in and repeat the whole HDX. You can do temperature dependence. You don't have to have your protein cold if it's happy to be a bit warmer. You might get different exchange kinetics and infer a different conformational behavior then at different temperatures. So you can push that quite far. but it's still a slow, expensive experiment. Very briefly, maybe I skipped this a bit more. There's so-called EX1 and EX2 kinetics, because depends on how fast the exchange is compared to the opening or closing kinetics. Typically, you look at EX2. You get a continuous shift of the isotope cluster. So this is 1 Dalton spaced apart, or if it's doubly charged, half a Thomson. But if you have EX1 kinetics, then you basically have a situation where it's more open, more of the time in an open state. And so then in a relative sense, the opening is not a rate limiting step anymore. So you might have seen something analogous in NMR if you're into that, but it's not quite the same, but it's an interesting regime where you can actually, if you have a more disordered protein, you expect potentially more of the EX1 kinetics, but it also has to be coordinated along the peptide. It's not just one residue, they all have to do it for that strict EX1 behavior to occur. So this is actually a complication for software as well. It's not very good at detecting this. And I should say that EX1, EX2 kinetics is often wrong, even in publications, because proper EX1, the one cluster goes down in intensity as the other one increases. This is not even the best cartoon because the total intensity should more or less be constant. You could have 2 clusters of the same peptide because the protein is just in 2 states, which coexist. That is different, so you have to be careful. Frontiers of HDX, and really exciting now because it's also becoming commercial. Millisecond HDX is important for intrinsically disordered proteins, simply because they exchange fast. There might still well be a difference, fast and faster. But if your first time point is after 30 seconds, then you pretty much, if you wait long enough, you know, all the accessible sites will be deuterium. So you can't see the difference. So you need faster sampling. Millisecond HDX. What we also need to understand better is salt effects. And we need to control our back exchange. We can't avoid it, but we ideally quantify back exchange. And there are models now where basically once you have digested your protein and you have these peptides, and they're then quenched, but there's still some low-level activity, you could model that part, and then you can actually maybe model back exchange in, because the true HDX data comes from NMR, where you don't have back exchange. But as you know, HDX NMR is not massively popular. We do it actually sometimes for reference, and you have to label, and you can't do it on very large systems, either on ribosomes or so. So it's a bit tricky, but if you want to calibrate your mass spec HDX, you can use HDX NMR data, and we've just got a paper in a pipeline submitted soon where we do that systematically and also look at how much more accuracy we get into predicted models from that. Just show this here briefly to, I guess, impress you a bit with what you can do. So this is a study with a colleague and also with a pharma company where we look at an APO enzyme and various cofactors and ligands and inhibitors. And what you see, even if you don't try to understand this in detail, you see one pattern which is slowly shifting, EX2 behavior, and a second one which is just appearing, EX1 behavior. So you actually get, even in the APO form, you get over the time of these 4 time points, you get a switch from one regime to the other. But if you then have any of these small molecule modulators, you see how that might still happen, but at in different time frames. So you can actually really get kinetic data on the inhibition from these kind of arrays of systematic experiments. So just to say that's in the power of HDX. An example for millisecond HDX from the colleague J.J. Phillips in Exeter, wild-type alpha-synuclein, and the phosphorylation is important in aggregates. You actually find it in Lewy bodies. And you can see now this required a millisecond HDX, and you don't see amino acids here, but peptides. But you can see certain regions where the peptides are differing a lot between the wild type and the mutant. So you can actually understand what part of the protein is really affected by phosphorylation, and is that driving the different aggregation kinetics. It's interesting now, increasingly also for higher throughput. There are, of course, attempts to make it quicker. Do we need all 5 time points is an important question. Can we go into cells? It can actually drive IDP drug design. You might maybe not have heard of a startup company, ultimately came out of the lab of the late Christopson, where they use just millisecond HDX for their IDP drug design. So really to make drugs for IDPs based on this. And you might have heard of Gabi Heller's Bind company, which tries to do something with NMR data, not so commercial, but also try to understand behavior of IDPs based on NMR data. There is some attempt on reweighting ensembles. I mentioned this, and maybe I skip through this a bit more. It's a paper to look at, and people have picked up on it now, but it's in this very limited scenario where you don't assume you know nothing, but where you basically look at two states. I just want to highlight here, this is also from a review article I showed on a previous slide, that you see the difference between, let's say, apo and ligand bound. So ligand bound state, it might have more deuterium uptake, it might have deprotected something. So that's when you compare two states. But what's equally interesting is, actually, do you get any incorporation, even if it's constant over time, like here, where you have the maximum incorporation or not? So you can actually even infer if a peptide is on the surface or not, even if you don't compare with ligand and without ligand. And that's something we really don't use much at the moment in HDX. So there is a recommendations paper, already also 6-7 years old. We're working on a follow-up, which focuses a bit more on software, because there still is also for HDX, and this is really a shortcoming. This is used much more widely now already. No structure depository. We are in advanced discussions with DRIIMB at EBI, so that's the proteomics depository. Some pharma companies have some in-house solution. They're also happy to share to the community because they're doing this quite systematically, and they want to drive some machine learning, actually not for structural predictions, for functional predictions. But important that metadata gets captured. Otherwise, you have different experiments and you think they're the same and they're not. Best practice isn't always followed, and the data analysis pipeline is painful. It takes twice as long to analyze the data than do the experiments. So a 1-week experiment turns into a 3-week exercise because we are still lacking much more streamlined, automated, smart software. There are pieces and bits, and manufacturers provide some, but it's not streamlined. More recently, we looked at all the software. There's already 1 or 2 more because this is now a 1.5 years old. But there is a lot happening, but it needs to be tied together properly. So if someone's interested in the software tools, then this is quite a nice summary. I'm sorry if I'm going to be a few minutes over. I come to the last techniques. Actually, this one is going to take a little bit longer. The last one will be very quick. Covalent labeling is a technique which is not used so much yet. It's actually using a chemistry, radical chemistry normally, where you do something a bit like HDX, but you make a permanent change to a side chain in a protein. So you're not exchanging a deuterium in an amide position. You can do that in principle even in cells and even in nematodes. A colleague in the US does that. But more typically, it's also done with isolated purified proteins. You can then look at docking again, like epitope mapping, but you can do interactomics. You can do it proteomics level wide. You can look a bit like HDX at structural transitions, IDPs. You can, I come to that later, use a laser synchrotron or plasma. And you can actually, and this is important, get to single amino acid or even better in terms of your spatial resolution. Data analysis workflows, actually, they're on the scale of even months, not weeks at the moment. So you only do that when you have a very important data set. When you want single amino acid resolution. When you want peptide level resolution, it is much more straightforward, but only recently. You need something to generate your hydroxyls. Some solutions can be put on a bench, a synchrotron not so much. Come to that later. You also want your sample not to be too heterogeneous because then you're labeling everything and you don't necessarily know which part showed what difference. What is it actually? So in HDX, you exchange HD in an amide of the backbone. In hydroxyl radical protein footprinting or fast photochemical oxidation of proteins or some other flavor of covalent labeling, you use an oxygen radical and it will modify something in your side chain. But if you look at this flow diagram here, you don't need to quench at acidic pH. You can use just to get rid of excess radical. You can use catalase and methionine reacts with a radical. You can then actually freeze your sample. You can put it through a normal trypsin proteomics workflow like a PTM map, which is a great advantage. So you're not going to lose your label as you do with the deuterium. And then you can look at what you produce, and you can also quantify 2 conditions where you see now this amino acid can label more because it's more exposed or because something else changed. But they're related yet different because in hydroxyl radical footprinting, covalent labeling, you're really getting data on the solvent accessible surface area. It's not predominantly, we don't think it's depending on the secondary structure in fact at all. So it's actually potentially easier for modeling, right? Because in HDX, you see a difference. It could be because that loop's moved out, or it could be because that loop was structured and is now unstructured. And you can't immediately say that. And here you can just say the SASA has changed. Now you might not see everything on the surface. It's partial data. But what you label, you can assume should be on the surface. And then the other advantage is you get covalent modifications versus labile in HDX. And you can use classic proteomics to find basically what's like a PTM, rather than having this inline pepsin fast workflow where you can't do complex samples because you do very quick LC, while here you can do your 2-dimensional 2-hour separation if you want. So it is more versatile, but it's also less common and more painful in terms of data analysis at the moment. That can change, of course. So we in Leeds were the first outside of the US, and now there's also in Prague, a second setup in Europe, to use a UV laser, so an excimer laser, and you can then very quickly on a microsecond time scale, you can flash a piece of your sample which comes to the capillary, or it can also be potentially on a surface in a tissue, and you get oxygen radicals, but it's sparse, so you're not oxidizing your protein much. It's actually just one of these which gets oxidized. And there are some amino acids which oxidize more easily than others. It's also not surprising, but in principle, all 20 of them can oxidize because it's radical chemistry. And you just find your oxidations later and you reconstruct a structure from that. But where the challenges come in is that you had an unmodified peptide before and it had one retention time in the liquid chromatography. And then you have just a single oxidation plus 16 on any amino acid and maybe even in more than one possible position. So then you have a couple of new peptides and your quantification of how much is still unmodified and how much is now plus 16 should be based on the chromatographic peak intensity, not on the mass spec peak intensity. It's good practice. But how do you align new peptides with the old peptide? That is a big challenge. At the moment, the answer is manually. But we are working on some AI predictions with a group in Belgium where they can predict how a PTM shifts retention time. So we like to automate that step using machine learning. A really nice example from, I guess, my predecessor in Leeds, Alison Ashcroft, and a PhD student then who now works at Waters and does HDX. So hydrogen-deuterium exchange compared to FPOP on an interesting protein, microglobulin, which also makes amyloid. I'm not going to go into too much detail, but in HDX, you have these time points and you see areas which change. In FPOP, you might just see some residues, but you could even see different isomers, orthometa para, on an aromatic phenylalanine. And depending on how this particular residue has turned, it actually tells you even the orientation. This is not always the case, but it's an example of sub-amino acid spatial resolution here. And you can see how blue and red, the two scenarios, wild type and mutant, how you can then quantify differences, but based on chromatography. It's fair to say that Owen worked for almost a year just on that particular study. But it was a proof of concept. So here's another example. If you also look for double oxidations, which do happen in such a peptide, you see how many new peptides you've made, which carry the oxidation in different positions compared to the unmodified peptide. And an oxidation normally makes a peptide, you would think, more polar. So on reverse phase, it should appear earlier, but that's not always true. So certainly histidine modifications and sometimes tyrosine and phenylalanine can also be later in retention time. But the machine learning and the colleagues in Belgium who've done this on lots of proteomics data, which is deposited at the moment already, they have a pretty good prediction tool now where they can kind of say this is likely to be that position because of the retention time even. But of course, in the mass spec, you can also, you get a plus 16, you can do some MS analysis. You can then confirm where the oxidation really went because it contains spatial information on the protein, right? But you can see that you can drive that quite far and it gets very complex. Very briefly, very recent work, not published yet even, is here in-house, but also with AstraZeneca. We've looked at AAVs, at whole adeno-associated viruses. We can also do native MS of them, by the way. But we are looking here with FPOP at the sort of differential patterns of labeling when we compare full and empty, but also we compare heterogeneous VP123 with homogeneous VP3 only capsids. And we can learn quite a bit about the additional sequence on VP1 and 2. We can learn something about the genome packing, not going into any detail, but FPOP allows us to find some highlighted areas of difference in labeling, which means difference in conformation and accessibility. So just to wrap that up, and then I'm almost done. So fast photochemical oxidation of proteins with a UV laser was invented by Michael Gross in the US. It's done by some other colleagues, mostly in the US. Also, some pharma companies already routinely use it. You can use diode lasers in a benchtop scenario, but we have the UV laser, which is more powerful, also useful for cellular labeling, which is only in Leeds and recently in Prague. So you can do this for bispecific antibodies and lots of interesting structural questions. You can actually use cold plasma and electric discharge as well. So a plasma generator, a small plasma pen can be used, but and maybe that's the point I want to make. X-ray footprinting at a synchrotron is happening at 2 sites in the US. Mark Chance is the person who really set this up, Brookhaven and Berkeley. But we are now working with our colleagues at DRIIMB to make a case, and we will submit that soon for funding to establish hydroxyl footprinting on the synchrotron in Havel. So then hopefully this will become something which can also be done outside of the US, which is important because we don't even know if Brookhaven might continue with funding and other things which are happening. So peptide level workflows are okay, but actually the nightmare, it is worth it though in some cases. It starts when you really want to pull out quantitative single amino acid level information, because there's not much of a good workflow yet. We know what needs doing. We could automate it, but it needs some serious programming and a bit of a more of a consensus what everybody agrees should be done. The stats is done. We do replicates and everything, so we're not just pulling out something we like. But we also like to have a bit more of a fundamental understanding of the radical chemistry. So what has a high activity under labeling conditions, so we can expect more what label we should see. And there is a potential for fast and efficient epitope mapping. So pharma companies might switch from HDX to this kind of approach in the next couple of years, because once the software and everything else works, it is actually more robust, quicker, probably cheaper than HDX. There is a very recent paper, so far it's on ChemArchive, but we hope Nature Methods will accept it very soon, where mostly the American colleagues did contribute to some first sort of peptide level qualitative epitope mapping type FPOP and synchrotron footprinting experiments to basically give a bit of guidance to the community. But this needs to be followed up with a more advanced analysis, and also we can pull out much more information from these data, as I showed. The last point I want to go through very quickly is cross-linking. The reason I want to do that quickly, chemical cross-linking, many of you might be quite familiar with. It's the most developed technique. So I went from the biggest challenge to the smallest. It had been used a lot. I have it here when DRIIMB was still blobology. So when you had low resolution data and you could dock your subunits to match the sort of envelope, and then cross-linking data. And I think it still plays a role for that, and was really useful because it gives you contact information. So it's a native labeling of sorts as well. In fact, you also get so-called dead-end cross-links, which don't cross-link, but just link to nothing. And they're actually like the covalent labels I talked about, right? But normally you look for the ones which connect 2 parts of the protein, either the same protein, intramolecular or intermolecular. It can be quantitative, but this is quite new. So people normally just said, oh, here's cross-links, so I think this is how they interact. But then there might be some other cross-links which don't match. And it can be that you have a heterogeneous scenario where either there are 2 states or it's a breathing, basically, and you don't capture that, even though it's in the data. So there are still challenges here as well. But it is relatively routine now for some modeling, because you can take a leaf out of the book of NOE effects in NMR, or maybe FRET, where you also have some distance information. You can cross-link with different length cross-linkers, and then it can't be more apart than the length of the cross-linker. So there's quite a lot of sophisticated stuff people have done, but it's still not, it's done when you confirm a model you have, right? It's still not done as routinely as I think it could be, because the idea is to do it in a whole proteome, and this is still a challenge. So predict interactions and map interactomes. So it's the most established structural MS method. It's the most modeling conform approach. And a lot of tools like the chemicals you use, which click into these positions on the proteins, you can buy. So companies have developed some protocols. It can be used by a non-mass spec expert, although it's still good to have one at hand, to be perfectly honest. But there are more protocols and kits nowadays for this. It's very important. But there's less to say here because it's more worked out already, I think. There's been a good sort of review again around 2020, came out of this so-called cost action, where the community also looked at reliability and so on in cross-linking. In fact, it turned out there was second paper, there was another one where we just said, take some BSA, bovine serum albumin, and cross-link it the way you like, and report the data anonymously back to us. And it was shocking how some people had 5 cross-links and some 200. So some standards and best practice were needed, and this is already a follow-up. It's still ongoing, but people do it quite a lot. So just to wrap up, sorry I'm over time, we have a toolbox in mass spec. You can do quite a lot. Some approaches read out a native scenario from a solution. Some use labels, then denature and digest, and then read out the labels. but you can see how you can reconstruct that into structures. And this is again borrowed from Thermo Fisher, where they also talk about cryo-EM and mass spec, how they work together. Bit of an idea how different types of binding interface, surface accessibility, and conformation shape information can feed together with cryo-EM to also get a dynamic picture of a protein complex. But in mass spec, it's often a game of where's Wally. I mean, if you have an oxidation in hydroxyl footprinting, you might have hundreds of proteins, and then you're looking for the one specific site which tells you something about where this happens. So unfortunately, this is still a challenge, but the mass spec technology is getting better as well. Want to thank the larger MS groups, so several groups actually working together on this in Leeds, and also some of the funders. And thank you for your attention, and hopefully you're not so exhausted that you still have some questions to ask. Thank you.
Okay, thank you. That was great. That was a great overview of the big five. So yes, we do have a couple of questions. Maybe I'll read them out, or hopefully you can see them as well, but let me read them out. So Eric was asking a question at the beginning. I think this refers to native mass spec, where you were describing the estimation of KD. He says, regarding the binding affinity measurement, did they correct for non-specific binding?
It's a very good question, Eric. I think you have the idea because you work in that field as well. We didn't here, but of course, there are good examples from various labs. If you want, I can later email you some, where people have done that. So I think a good practice is to look for non-specific binding as well, control with a protein which shouldn't bind. I did show it for the aptamer though. There we did that, yeah.
Well, Eric, you can speak now, is it?
Yes, at some point they unlock that.
Okay.
Thank you.
Sorry, I should have let you ask the question. Okay, and in the Q&A, there's a question from Hassan on native mass spec. Are you able to ask it yourself, or shall I read it out?
Yeah, I think we have to read it out.
Yeah. Okay.
Mike's.
So, so native mass spec, is nanoflow the mainstream and microflow is only used occasionally? What is the minimum concentration of ammonium acetate that can be used? And is there an educated guess to choose the optimal concentration, or is it more empirically inferred?
So it's a very good question as well. I think I did talk briefly about how in native MS we had some limitations with having to use volatile buffers, and even though we can add some magnesium if we needed an ATP, we limit it. But I mean, Eric, who just asked the question, had a very nice recent paper as well from his work, how we can use now the kind of glass needles which are so fine that you make your droplet mist in electrospray. You make it actually not even nanospray, you make it picospray now, and you get a much better tolerance because your droplets are so much smaller. You don't get that sort of competition with maybe surface active detergent and salt. The question was about the minimum concentration. So my point here is now that we want to keep the protein happy. So if someone does a gel filtration purification, which is still not uncommon if you prepare protein for any structural technique, then we look at that, right? And we look at maybe the glycerol and say we don't need that because we're not freezing. But if it needs high salt, like some kinases are more stable in high salt and protein DNA, you don't want it too high, and we match it. And we normally match it with ammonium acetate, but we now see we can actually even study sodium versus potassium, for example, if we think there is an effect. So this has become less of a limitation, but you do want some conductivity in your solution for electrospray, but actually the protein wants something as a buffer, right? It wants some salt of some sorts. So I think that's actually what we try in the first instance to match. And now we can do that more than before because we realized we can use more of a non-volatile buffer than we thought we could by using these much finer tips. The protein concentrations, by the way, for native, they're in the low micromolar or slightly sub-micromolar concentration, so roughly like a cryo-EM experiment or maybe like an X-ray experiment. But we might need a bit more to prepare the sample to load a bit more, but we're not consuming much. I think that covered more or less what you asked.
Yes, so okay, thank you. We have one final question written down. From Sarah, I'm not sure which Sarah this is. It could be the Sarah. Can computer simulations that mimic the experiments, i.e. digital twins, help with getting more from MS data, and should we invest in developing them?
I think that's a great idea, Sarah. And there's one group in particular which has done a, let's call it digital twin to the whole electrospray process. And that actually has helped us a lot to understand how these interplays, and particularly the interplay of charge state and conformation of solvent accessible surface area, how that works. And I mean, we have even, I suppose, more or less conferences which focus on biomolecular structure in solution versus gas phase, because a lot of optical spectroscopy started out in gas phase scenarios. So when we worry, which we should always, how far bringing this structure into the vacuum could change something, and we need to know that, right? If we know it, we can control it. Then that kind of digital twin of the electrospray process can be very helpful, particularly for native. The moment we label, a lot of these problems go away, but it's kind of the fact that both approaches, both families of structural approaches have their unique advantages and disadvantages. I like to also remind people, but of course you know that at cryo-EM, even though you're kind of protecting with a vitrified liquid layer, you're kind of in vacuum. I mean, there's certainly, if you don't have cryo, you have issues with a vacuum. And in X-ray, I know a bit myself about crystallization artifacts. So I think any technique, maybe NMR the least, although NMR has to use higher concentrations, you have some potential issues that might not be major, but you need to really understand them. And mass spec's always been very much aware of the fact you go into the gas phase and you're not folding in the gas phase. That's important to say, right? Which is preserving the structure if you're lucky and do it right.
Okay,
but because a lot of these techniques are quite recent, we're definitely lagging on help from the computation community. We can sometimes borrow things which has been, maybe something's been developed for SACS, you know, or for some other technique. So I think it's good to intensify the dialogue.
Absolutely. So yeah, can I ask one question for myself? Just thinking, just want a general question as to whether there are size limits in terms of the size of the complex that you look at. And I think I'm thinking mainly here about HDX and covalent labeling.
Very good point. I think that's actually an asset of the technique. So colleagues in France and also colleagues at the DRIIMB have looked at things like proteasomes and ribosomes and really at the whole particle in HDX to see how a particular specialization, so when a subunit gets replaced by a different subunit, specialized ribosomes, also proteasomes in different sort of types of cells can vary a bit in their composition. And so you can really look at those peptides in context, because you can then, in the end, even if you have 50 proteins, like you might have on a ribosome, you just have to, when you digest this down, you have to look for many more peptides, so your sensitivity has to be good. But these things are done also on nascent chain ribosomes at a quick. And with native MS, we can definitely look at whole ribosomes. It was quite a breakthrough when we first managed to do that with Carl Robinson. And we managed to, I guess, tweak the instrument to be good in a megadalton range. But now we look at sometimes even larger virus capsids than AAVs. I think native is capable of it. Covalent labeling definitely is capable of it in HDX as well. You do get a bit of a challenge with having more peptides and needing more sensitivity. But this is not a major challenge because you kind of just potentially have to use a bit more sample in the end, right, to make up for the sensitivity challenge. But I think it's a big asset. And you can look at small particles with membrane proteins in them. You can have different lipid compositions maybe in a nanodisk where you can manipulate it. And you can see how the transporter or your membrane protein responds to a different lipid environment with HDX, for example. While with native, you would look at the whole particle. You don't want to look at the lipids, right? So you would have to peel it out somehow. There are some tricks for that as well. But with HDX, it's not even an issue per se. And there are good papers published on this by Argyris Politis, for example, Chloe Martins.
Okay, yeah, thank you. That sounds very encouraging. Are there any other questions, either from people who can speak or people who can type into the chat or the Q&A? I think not. So yeah, so So you were happy for this to be recorded. We're going
Yes.
to make this available because this strikes me as an extremely useful reference for those of us new to this.
Yeah, I'm happy with
Yeah,
that. And it's very much work in progress. I mean, I'm sure I missed out on a lot of exciting things people have tried recently, but I wanted to set the stage for like an engagement of our community, which I guess is overlapping with computational scientists, and we use some other techniques sometimes, but I think we can get a lot more done if we integrate more both ways. We consider mass spec more as a technique which can do partial structural information, not XYZ coordinates in the same way as other techniques, but can contribute. And I think we can learn a lot from looking at what developments happened, CCP4 going back a long way, more recently NMR and EM as well, and BioSim, of course.
Yeah, I mean, you made a lot of relevant comments about standardization and software pipelines and robustness. And, you know, these are, I think we've all had these experiences. And,
exactly, yeah.
Yeah, it's very important to work together.
Yeah. Yeah,
okay,
thank you very much.
Yeah, thank you very much for a great talk. Bindu, are we going to advertise next week?
So it's by Andrew French on AI in the biosciences. So it's just 1 week away, but yeah, hope everybody from the community can join that.
If I may also make an announcement, if Sarah Harris agrees, we are planning, so it's not fixed yet, there's no date yet. We are planning to do some kind of event via CCP BioSim where we want to dive a bit deeper into what we can do with mass spec data and invite speakers to that as well. So that's planned, but I'm sure through your channel it will also be announced. So if Sarah wants to say something or if she's here, but we hope to do that in the summer. I think we said potentially late June.
Yeah, I'm sure we will advertise that widely when it's organized.
Yeah.
Okay, I think we have run over quite a bit, so I think
yes.
we should wrap it up there. But yeah, thanks again, Frank, for a great talk.
Thank you for the opportunity.
Thanks to everyone for joining.
Yeah. Okay. Thank you.
Bye.
Thank you all.
Okay, bye all.
Bye. Bye.
Bye.
Thanks, Martin.